lymphocytes, activation and mechanism of action. Like B cells, T cells are also subject to a process of clonal selection. T cells possess a cell surface protein called the T cell receptor that allows them to interact specifically with a particular antigen. Like the antibody molecules that serve as B cell receptors, the proteins that serve as T cell receptors exist as a large population of molecules that have differently shaped combining sites. Just as each B cell produces only one species of antibody, each T cell has only a single species of T cell receptor. It is estimated that adult human possess approximately 10 raised to 12 T cells that collectively exhibit more than 10 raised to 7 different antigen receptors. T cells are activated by fragments of antigens that are displayed on the surface of other cells called antigen presenting cells or APCs. Once again, T cells are activated by fragments of antigens that are displayed on the surface of other cells. So consider what would happen if a liver or kidney cell were infected with the virus. The infected cell would display portions of the viral protein on its surface, enabling the infected cell to bind to a T cell with the appropriate T cell receptor. As a result of this presentation, the immune system becomes altered to the entry of a specific pathogen. The process of antigen presentation is discussed at length later, and whereas any infected cell can serve as an APC in activating T cell, certain types of professional APCs are specialized for this function. So professional APCs include dendritic cells and macrophages. We will focus on dendritic cells, which were first discovered and characterized by Ralph Steinman of Rockefeller University in the early 1970s and are often described as the sentinels of the immune system. Dendritic cells have earned this title because they stand guard in the body's peripheral tissue where pathogens are likely to enter, such as the skin and airways. So, dendritic cells are equipped with a wide variety of receptors capable of recognizing virtually every type of pathogen. This property makes disease a major component of the innate immune system. So, disease in the sense dendritic cells utilize the information about the pathogens they ingest to initiate a response from the appropriate cells of the adaptive immune system. So, when present in the body's peripheral tissue, immature disease recognize and ingest microbes and other foreign materials by phagocytosis. Once a microbe is taken into a dendritic cell, it has to be processed before its components can be presented to another cell. So, antigen processing requires that the ingested material is fragmented enzymatically in the cytoplasm and fragments move to the cell surface. Tissues that have processed antigen migrate to nearby lymph nodes where they differentiate into mature antigen presenting cells. Once in a lymph node, disease come into contact with a large pool of T cells, including a minute percentage whose T cell receptors can bind specifically with the processed foreign antigen which activate the T cell. This dynamic process of DC-T cell interactions has been visualized recently in living lymph node tissues by microscopic imaging of fluorescently labeled cells. In the absence of antigen, a given DC may interact transiently with as many as 500 to 5000 different T cells remaining in contact with each cell for only a few minutes. So, in contrast, when the disease display an antigen that is specifically recognized by the TCRs of T cell, the interaction between the cell is seen to last a period of hours, leading to the activation of T cell, as evidenced by a transient increase in the cytosolic Ca2 plus concentration. So, once activated, a T cell proliferates to form a clone of T cells having the same T cell receptor. It is estimated that a single activated T cell can divide 3 to 4 times per day for several days, generating a tremendous population of T cell capable of interacting with the foreign antigen. The massive proliferation of specific T lymphocytes in response to an infection is often reflected in the enlargement of local lymph nodes. Once the foreign antigen has been cleared, the vast majority of the expanded T cell propulsion dies by apoptosis, leaving behind a relatively small population of memory T cell capable of responding rapidly in the event of future contact with the same pathogen. So, unlike B cells, which secrete antibodies, T cells carry out their assigned function through direct interaction with other cells, including B cell, other T cells, or target cells located throughout the body. This cell-cell interaction may lead to the activation, inactivation, or death of the other cell. In addition to direct cell contact, many T cell interactions are mediated by highly active chemical messengers called cytokines that work at very low concentrations. So cytokines are small secreted proteins produced by a wide variety of cells and includes interferons, IFNs, interleukins, ILs, and tumor necrosis factors or TNFs. Which are the cytokines are include interferons, interleukins, tumor necrosis factor. Cytokines bind to specific receptors on the surface of a responding cell, generating an internal signal that alters the activity of the cell. In responding to a cytokine, a cell may prepare to divide, undergo differentiation, or secrete its own cytokines. So, one family of small cytokines called chemokines act primarily as chemoattractants that stimulate the migration of lymphocyte into inflamed tissue. So, different types of lymphocytes and phagocytes possess receptors for different chemokines so that their migration patterns can be separately controlled. Some of the major cytokines have given IL-1, diverse sources are there. Then major functions include induces inflammation, stimulates Th cell, and along with that proliferation will be taking place. Then IL-2, Th cells stimulate T cell and B cell proliferation. Then IL-4, T cells are the source. Then induces IgM to IgG class switching in B cells, suppresses inflammatory cytokine action. Then IL-5, Th cells secrete them. Then stimulates B cell differentiation. Then IL-10, T cells macrophages, inhibits macrophage function, suppresses inflammatory cytokine action. Then IFN gamma, Th cells, CTLs, induce MHC expression in APCs, activates NK cells. Then TNF alpha, diverse, induces inflammation, activates NO production in macrophages. Then GM-CSF, Th cells, CTLs, stimulate growth and proliferation of granulocytes and macrophages. Then three major subclasses of T cells can be distinguished by the proteins on their surface and their biological functions, which are the cytotoxic T lymphocytes. Screen these cells of the body for abnormalities. Under normal circumstances, healthy cells are not harmed by cytotoxic T cells, but aged or infected cells and possibly some malignant cells are attacked and killed. So, cytotoxic T cells kill target cells by inducing them to undergo apoptosis. Two distinct cell killing pathways have been described. In one pathway, the CTL releases perforins and granzymes into a tightly enclosed space between the cells. So, cytotoxic T cells kill target cells by inducing them to undergo apoptosis. That is one method. Two distinct cell killing pathways have been described. In one pathway, the CTL release perforins and granzymes into a tightly enclosed space between the cells. So, perforins are proteins that assemble within the membrane of the target cell to form transmembrane channels. Once again, I'm going to read. Cytotoxic T cells kill target cells by inducing them to undergo apoptosis. Two distinct cell killing pathways have been described. In one pathway, the CTLs release perforin and granzymes into a tightly enclosed space between the cells. So, perforins are proteins that assemble within the membrane of target cell to form transmembrane channels. Granzymes are proteolytic enzymes that enter the perforin channels and activate caspases, which are the proteolytic enzymes that initiate the apoptotic response. I'm not listening. So, once again, I'm going to read this because I'm confused. Cytotoxic T lymphocytes. screen the cells of the body for abnormalities. Under normal circumstances, healthy cells are not harmed by CTLs, but aged or killed. Infected cells and possibly some malignant cells are attacked and killed. You know that. Then, cytotoxic T cells kill target cells by inducing them to undergo apoptosis. So, two distinct cell killing pathways have been described. In one pathway, the CTLs release perforins and granzymes into a tightly enclosed space between the cells. So, perforins are proteins that assemble within the membrane of the target cell to form transmembrane channels. Granzymes are proteolytic enzymes that enter the perforin channels and activate caspases, which are the proteolytic enzymes that initiate the apoptotic response. In the alternate pathway, the cytotoxic T cell binds to a receptor on the target cell surface, activating a suicide pathway in the target cell similar to that. So, by killing infected cells, CTLs eliminate virus, bacteria, yeast, protozoa, and parasites after they have found their way into host cells and are no longer accessible to circulating antibodies. So, CTLs possess a surface protein called CD8 cluster designation 8 and are referred to as CD8 plus cells. Then helper T lymphocytes. Th cells activate other immune cells. Once again, helper T lymphocytes. Th cells activate other immune cells in an orchestrated attack against a specific pathogen. They are distinguished from cytotoxic T cells by the presence of the protein CD4 at their surface rather than CD8. Th cells are activated by professional antigen presenting cells such as dendritic cells and macrophages. This is one of the first and the most important steps in initiating an adaptive immune response. Nearly all B cells require the help of Th cells before they can mature and differentiate into antibody secreting plasma cells. So, B cells are activated by direct interaction with the Th cell that is specific for the same antigen. Thus, the formation of antibodies require the activation of both T cells and B cells capable of interacting specifically with the same antigen. The importance of Th cells become apparent when one considers the devastating effects wrought by HIV, the virus that causes AIDS. Th cells are the primary targets of HIV. So, most HIV infected people remain free of symptoms as long as their Th cell count remains relatively high, above 500 cells per ml of blood. The normal count is over 1000 cells per ml. Once the count drops below approximately 200 cells per ml, a person develops full-blown AIDS and becomes prone to attack by viral and cellular pathogens. So, Th cells are very important. Okay. Then regulatory T lymphocytes. Regulatory T lymphocytes. T regulator cells are primarily inhibitory cells that suppress the proliferation and activities of various types of immune cells. So, virtually all of the suppressive activities of T regulator cells have been studied in vitro, so it remains much less clear how these cells operate in the body. So, Treg cells are characterized by possession of CD4 plus CD25 plus surface markers and are brought to play an important role in limiting inflammation and maintaining immunologic self-tolerance. So, Treg cells carry out this later activity by suppressing CTLs that carry self-reactive receptors from attacking the body's own cells. So, Treg cells appear also to protect a fetus from immunological attack by the pregnant mother. On the other hand, the same cells may prove detrimental to our health by preventing the immune system from ridding the body of tumor cells. The differentiation of Treg cells requires stimulation by the cytokine IL-2 and lead to the expression by the cells of a key transcription factor FOXP3. Mutations in the FOXP3 gene result in a fatal disease IPEX which is characterized by severe autoimmunity in newborn infants. Defects in T regulator cells are widely suspected for playing a key role in the development of most autoimmune diseases. So, studies of Treg cells have provided direct demonstration that homeostasis in the immune system require a tight balance between stimulatory and inhibitory influence.